Hi, am I audible? Yeah, okay. So today is our tutorial for the 10th week of Animal Physiology, and this has been postponed from Saturday. I am Gitika Agarwal. I'm a PhD scholar at the Center for Ecological Sciences, IIC Bangalore. And today we'll be talking about what was discussed in the 10th week of Animal Physiology, and let's keep it an interactive session. If you guys have any doubts, you can stop me in between whenever you want. Okay. So during this week, mostly what we talked about was about the anatomy of the heart, the structure of the heart, and then we looked at how blood flows in the heart and in the cardiovascular system, how there are two systems that are at play for this flow of blood, which is the conduction system and the contraction system, how these both work in the heart, and what are the different types of cells that are present in the heart and that form these different conduction and contraction systems. So, coming to the heart, so can you guys tell me about what are the four different chambers that are present in the heart and what are the different valves that are present in the heart? So this is a basic diagram of the heart you can see here. So can you tell me what are these four different chambers that are found in the heart and what are the different valves? So you can see that there are two chambers in blue, two in red, and there are four types of valves in white. So can you tell me what are these different chambers of the heart? What is the one that you can see here? Can you see my pointer? What is this chamber? Anyone? What are the four chambers of the heart? What are they called? LARALVRV. So what does the A and the V here stand for? Okay, you're saying it's right oracle. So are you sure? Can you think of anything else? It's not oracle. So what do the A and the V stand for? Atrium and ventricle, exactly. So it's the so we have these four chambers. Two of them are the atria and two are the ventricles. We have the right ventricle in the right atria and the left ventricle in the left atria. So, and what are the four walls? What are the four walls called? Excuse me, just give me 1 second. Hi. So, okay. So you're saying bicuspid, tricuspid walls, and the semilunar walls. Okay. So which ones are the semilunar walls? Are they ones that separate the atria and the ventricles, or they are the ones that are present at the openings of the veins and the arteries? So, what are these two walls? Are these the semilunar walls or are these the semilunar walls? Do the semilunar walls separate the atrium for the from the ventricle, or are they the ones that are present at the opening of the veins and the arteries, opening of the pulmonary vein and the aorta? Between the left ventricle and the aorta. So correct. So basically, the semilunar walls are the ones that are present at the openings of the veins and the arteries, and the other two that separate the ventricles from the atria are the bicuspid and tricuspid valves. Out of this, the one separating the right atrium from the right ventricle is the tricuspid valve, and the one separating the left atrium from the left ventricle is the bicuspid valve. Okay, fine. So now that we know about the basic anatomy of the heart, so, as we discussed, there are four heart chambers in the human heart, two upper chambers and the two lower chambers. Upper ones are called the atria, lower ones are the ventricles, and based on which side they're present on, there are the right atrium, right ventricle, left atrium, and left ventricle. And as we just discussed, there are these four valves. The one at the opening of the pulmonary vein is the pulmic valve. The one at the opening of the aorta is the aortic valve, and then we have the mitral valve or the bicuspid valve and the tricuspid valve. So, as you said, these are called the semilunar valves, the two that are at the opening of the vein and the artery, pulmonary valve and the aortic valve, and the two here at the atrioventricular junction are the tricuspid valves in between the right atrium and the right ventricle, and the bicuspid or the mitral valve at the entrance of the left ventricle in the left atria. Can you tell me why are they called tricuspid and bicuspid? What is the difference between tricuspid and bicuspid valves? Anyone? Why are how is the tricuspid valve different from the bicuspid valve? Exactly. So basic thing, you can sense it from the name itself. The tricuspid valve has technically three flaps that close the valve, and the bicuspid one has only two flaps that closes it like a normal door. Okay, moving on from here. So here is a GIF in which you can see how blood flows in the human body. So basically, from here you can see that blood enters into the right atrium, or the deoxygenated blood enters into the right atrium from the rest of the body, either from the superior part of the body or from the lower organs. It enters the right atrium, and from here it passes on to the right ventricle, and from here the pulmonary vein takes it to the lungs. Here the blood gets oxygenated because we are breathing, we're taking in oxygen. So the blood gets filled with oxygen and it comes back to the left atrium. From the left atrium, it passes on to the left ventricle. And from the left ventricle, it goes via the aortas to the upper and the lower part of the body. And again, this is a closed loop. So that is why it's called a closed loop system, because there is no loss of blood. However much blood enters into the heart, the same amount goes back to throughout the body. So it gets circulated throughout the body and there is no loss of blood in this system. It is a closed loop system. You can see here there is one part which is called the pulmonary circulation and the other part which is called the systemic circulation. So in the pulmonary circulation, as I said, the deoxygenated blood that has come into the heart at the right atrium, it gets passed on to the right ventricle, and from the right ventricle the pulmonary artery takes the deoxygenated blood to the lungs. Here it gets oxygenated and comes back via the uh, pulmonary vein to the left atrium. So this is the pulmonary circulation where the blood goes towards the lungs and comes back from the lungs. And the rest of the circulation where the oxygenated blood goes to the rest of the body and comes back as deoxygenated blood is called the systemic part of the uh, circulation. In this, you can see that in blue there is oxygen poor blood or deoxygenated blood, and the red one is the oxygen rich blood. Again, here is another diagram that shows how the right atrium receives the deoxygenated blood, which is marked in blue from superior vena cava and the inferior vena cava. It moves on to the right ventricle. From the right ventricle, pulmonary artery takes it away to the lungs. From the lungs, it comes back by the pulmonary veins to the left atrium, goes to the left ventricle, and then it gets circulated throughout the body. Okay. So, moving on to the blood vessels. So, a major role you can see in this circulation is played by the blood vessels, because the heart is the one that's pumping the blood, but the blood vessels are the one that are ferrying or taking the blood away from the heart towards the different body parts, and then from the body parts back to the heart. So, in the transport and the movement of blood throughout the body, blood vessels, they're the ones that are playing the major role. And they can be categorized at either arteries, veins, or capillaries. As you would know, arteries are the ones that carry the blood away from the heart, veins carry the blood towards the heart, and the capillaries are the ones that connect these two. And the largest artery is the aorta, which leaves the left ventricle and supplies the blood to the entire body. And the largest vein is the inferior vena cava, which brings the blood back from the lower organs, that is, from the legs and from the appendages, hands, back to the heart. And that is the inferior vena cava, which is the largest vein. Superior vena cava is the one that brings the blood back from the upper parts of the human body. So, the major differences between arteries and the veins is, as I already mentioned, the arteries, they carry blood away from the heart. The veins, they carry blood towards the heart. And arteries mostly carry oxygenated blood, except for the pulmonary artery, because that is one artery that carries the blood which is deoxygenated towards the lungs. Similarly, veins carry deoxygenated blood except for the pulmonary veins, which bring oxygenated blood towards the heart. Arteries, as you would know, have a high pressure. They have thicker walls because the blood is getting pumped into the arteries by the ventricles. And the veins, which bring blood back to the heart, they have a lower pressure and they have thinner walls. Another major difference is that arteries do not have valves, but veins, they have valves present in them. Can anybody tell me why is so? Why do arteries not have valves? How is blood being propelled there? And why do veins have valves present in them? How do these help uh, in the propulsion or the movement of blood through the veins? Can you tell me? Can anybody take a guess? Why do arteries not have any valves but veins do? In the arteries, the blood flows with the high pressure. Okay. So, what about the veins? Why are, like, what is the function of the valves? How are they helping? Okay, so the major reason why walls are present is, as you mentioned, okay, fine, in the arteries, the blood is flowing with high pressure. So that ensures that there is no backflow of the blood, because there's already a lot of pressure from the behind. So blood is getting propelled forwards. But in the veins, what is happening is, since the veins are carrying the blood towards the heart, and a lot of the time they are working against gravity. When they're bringing the blood back from our hands, from our legs, they're working against gravity, and that is why these walls are present, because at each point when the blood crosses the wall, these walls close, and they prevent the backflow, as Kamlesh is mentioning here. They prevent the blood from going back to the organs and keep it moving forward. So that is why these walls are present in the veins, but arteries don't need the walls. Okay, moving on from here. So, movement of blood through the heart. So we've seen that there is one type of circulation called pulmonary, the other is systemic. That is throughout the body. Now let's see how the blood is moving through the heart. So can you tell me? So during these lectures, they were discussed that there are these four events that happen for the movement of blood to happen through the heart. So can you tell me what are these four events that happen in the heart and that show how the blood is getting move how the blood is moving through the heart? So there are these four events that were outlined. Can anybody tell me what are these? Again, in this diagram, you can see that there is, yeah, okay. So the first event is the entry of blood in the right atrium, correct. Next.
Blood moves from left atrium to left ventricle through bicuspid valve.
Okay, from the left atrium to the left ventricle. Fine. So before that, what happens is a second event, which is almost simultaneously, the blood also enters the left atrium. Okay. After that, it actually blood moves. In the third event, the blood goes from the right atrium to the right ventricle, from where it goes to the lungs. So that is the third event. And the last is what you just mentioned. Blood goes from the left atrium to the left ventricle, and then it gets transferred towards the body. So you can see here, that's the first event, the blood which is entering the right atrium. And then blood enters the left atrium. Blood moves on from the right atrium to the right ventricle and then moves on to the lungs. And this the last one is that from the left atrium, blood goes to the left ventricle and moves on to the rest of the body, to the systemic circulation, basically. So you can see here in this graph, if these are these four events, they are all overlapping each other, mostly, more or less. So before the first event comes to an end, second event has started, and before second event comes to an end, the third event has started. So all of these events are sort of overlapping each other. So that is one thing to keep in mind. They are not separated from each other in time, they are overlapping each other. The next thing is in this circulation of blood through the heart, there are two major systems that play a role. One of them is the conduction system, which is also the electrical system, which basically controls these impulses that are going through the heart, which signals the contraction or relaxation of muscles. So the conduction system is the one that maintains what frequency these events are happening at, and because these events happen at a constant frequency, so conduction system plays a very important role in maintaining this constant frequency of these events. The second one is the contraction system, which sort of responds to the conduction system and the impulses that are received from the conduction system. And this is a mechanical system which carries out this contraction and relaxation, leading to the filling and unfilling of the heart chambers. So, overall, the heart works on an electromechanical basis. So, there is an electrical and a mechanical component to the pumping of blood through the heart. Now, let's get further and discuss this conduction system. So the conduction system is made up of the pacemaker cells. And you can see in this image, basically we have four types of pacemaker cells. We have a sinoatrial node, which connects to the atrioventricular node, also called the AV node, which then connects to these bundle of His. And these bundles are present in this wall, which separates the two ventricles. And then this moves on to the Purkinje fibers. Okay, in this diagram, you can see the exact location of these different pacemaker cells. There is this sinoatrial node, the SA node, which connects or sends signals to the AV node. From AV node, the signal moves on to the bundle of His, and from the bundle of His, it goes to the Purkinje fibers. And here you can see how it actually looks like. So, if this is the heart muscle in the pink, there is one layer of these pacemaker cells that moves through them, and when there is a signal passing through the pacemaker cells, the impulse also spreads towards the heart muscles. Another important thing to keep in mind is that these cells are at a resting membrane potential of negative 40 millivolts, which is different from rest of the neurons or rest of the cells that we have discussed earlier in these lectures, which were mostly at minus 70 millivolts. These cells are generally at a resting potential of minus 40 millivolts. That is why they are able to spontaneously shoot action potentials. They are active and they can spontaneously keep shooting action potentials, and they can keep firing these action potentials. They do not need an external signal to help them fire an action potential. So, this is the importance of the different pacemaker cells. Now, let's look at this short video which will explain how the conduction system actually works in the heart. So, kindly let me know if the audio and the video both are working for you guys.
The cardiac conduction system consists of the following components: the sinoatrial node, or SA node, located in the right atrium near the entrance of the superior vena cava. This is the natural pacemaker of the heart. It initiates all heartbeat and determines heart rate. Electrical impulses from the SA node spread throughout both atria and stimulate them to contract. The atrioventricular node, or AV node, located on the other side of the right atrium near the AV valve. The AV node serves as electrical gateway to the ventricles. It delays the passage of electrical impulses to the ventricles. This delay is to ensure that the atria have ejected all the blood into the ventricles before the ventricles contract. The AV node receives signals from the SA node and passes them onto the atrioventricular bundle, AV bundle, or bundle of His. This bundle is then divided into right and left bundle branches, which conduct the impulses toward the apex of the heart. The signals are then passed onto Purkinje fibers, turning upward and spreading throughout the ventricular myocardium.
So, was this audible? Did you understand what was going on? So, basically, what is happening here is that from here, from the SA node, the signal goes on to the AV node. And during this time, when the SA node sends a signal, the atria contract and they pass on the blood to the ventricles. And when the AV node then sends a signal to the bundle of His, then the ventricles start to contract. And eventually, when the signal reaches the Purkinje fibers, the ventricles entirely contract and they propel the blood to the blood vessels, to the pulmonary veins, and to the uh, aorta. So, is it the right or the left auricle? It's actually in the right auricle, in the light atrium. Okay. So the AV node is present in the, here you can see in the image as well, it's in the right atrium. And in the video also, you can see that it was shown in the right atrium. Okay. So moving on, coming to an electrocardiogram. Electrocardiogram is basically, you would have seen doctors studying the heart waves. So this is how the different impulses are traveling through the heart, and you can actually see if everything is working correctly in the heart by studying an ECG, or which is also called an EKG. So there are these different parts of an ECG. One of them is called a P wave. Then there is this flat segment called the PR segment. Then there is this QRS complex. Then there is this ST segment. And then there is this T wave. So these are the different components of an electrocardiogram. And when we look at them closely, what happens during the P wave is that the atria get depolarized. So, as we looked in the video, so the atria, before they contract, they get depolarized. So, during this depolarization, we see the P wave. When the SA node sends a signal, the atria they get depolarized, and that is when we see the P wave. And about 100 milliseconds after that, the atria contract and the blood goes into the ventricles. So that's just a small difference of 100 milliseconds. It's also called the atrial systole. So atria they contract and they force the blood into the ventricles. Then there is this QRS complex. During the QRS complex, the ventricles they get depolarized, and eventually during this ST period, these ventricles contract and send the blood ahead into the pulmonary vein and the systemic circulation. So this is called the ventricular systole. The ventricular contraction, it pushes the AV valves, they get closed. Both this tricuspid, bicuspid valve, both of them get closed. And in the second waves, the semilunar walls open and the blood is ejected into the uh, blood vessels. Eventually, in the T wave, what happens is the ventricles are getting repolarized, and after that, they get relaxed. And that is when these semilunar walls they close, and the AV walls they're still closed, but the blood is flowing into the atria. And then we'll come back to the P wave again. So these are the different components of a ECG wave. Here you can see a small GIF in which you can see how this is happening. So basically, when the P wave is happening, the atria they contract, they send blood into the ventricles. Then during the QRS complex, the ventricles they are contracting and they're sending blood into the uh, blood vessels. And during the T wave, the ventricles are both relaxed and the atria get filled eventually. So I hope this was clear how an ECG works and how these different impulses are studied in this wave diagram. Does anybody have any doubts with how to read an ECG or what is happening? Is it clear? Should I move on? Okay. If this is clear, let's move on. Uh, okay, you can't see the. One second. Can you see my slides? Oh, I will have to rejoin. Hi, sorry, the call got disconnected. Yeah, so here we were. Moving on from the ECG, we will look at how the action potential goes and how this is getting transmitted in the pacemaker cells and how it is happening in the cardiomyocytes, which are the which are the muscle cells of the heart. So first of all, let's look at how the action potential happens in the pacemaker cells. So now that we have seen uh, that the SA node actually sends a signal to the atria for them to contract, right? So that is what is happening here during the first phase. So all of the cells here in the blue line, they're actually repolarizing back to minus 60. So what happens is in the pacemaker action potential, there are three types of what do you call them? There are three types of channels: sodium channel, which are voltage gated, calcium channel, and potassium channel. All of these channels are voltage gated. So, what happens is for the sodium channels to open up, the voltage needs to be around minus 60 millivolt. And as you can see here, what is happening when the cells are getting repolarized, they're coming back to minus 60 only. So when they reach minus 60, when they hit minus 60, these sodium channels they begin opening, and this leads to an influx of sodium ions, and this is called a prepotential. So this influx of sodium is a very slow process, and by this slow influx of sodium, what is happening is the cell is getting depolarized because the potential in the cell is now increasing, it is getting less negative. Once the cell reaches minus 40 millivolts, that is a cue for the calcium channels to now open up. So this calcium channels, which are voltage gated, these now open up and there is a rapid influx of calcium. So when there is a rapid influx of calcium, you can see here by the red line, there is a sudden increase in the membrane potential and it goes to the positive side. It overshoots zero. When it goes above zero, that is when the potassium channel start opening. And as you know, potassium channels they lead to an efflux or an outflux of potassium. The potassium starts moving out of the cell. So you know all this already from the muscles action potential that we have studied in earlier lectures. So then there is a sudden outflux of potassium channels, and calcium channels have already closed when the potential goes to positive, and potassium channels open and there is a certain outflux of potassium, and this leads to a drop in the membrane potential back to minus 60, and that is again how this cycle starts again. So, an important thing to keep in mind is that there is nothing external happening to these pacemaker cells. There is no external stimuli. Itself, the cell goes back to minus 60. The minus 60 leads to opening of the the sodium channels, and then the cell goes back to a positive potential and then comes back to minus 60. So, there is nothing external driving this process. And that is why the our heart keeps beating, and there is no requirement of an external stimulus to this, because if there was an external stimuli requirement, you would need a constant stimuli for it to happen on a constant basis. Any doubts in this slide, or should we move forward? A few important points mentioned here in the red box are that the sodium influx is a slow process. Why? So that the ventricles get completely empty. It ensures that the blood is entirely flown out of the ventricles. The calcium influx is a fast process. It leads to the depolarization, causes the atrial contraction, and that is when the blood flows from the atria to the ventricles. And then repolarization, it resets the membrane potential so that another cycle of blood flow can happen through the heart. So, are there any doubts at this slide or in understanding the pacemaker action potential? If not, then we'll move forward. Okay. So, moving ahead, now that we have seen how the pacemaker cells are working, how these cells, the electrical system of the heart is working, now let's go to the mechanical part of it. How is the cardiac muscle themselves working? So, what is happening there? So, the action potential of the cardiac muscle, as you can see here, it looks much different from how the pacemaker action potential looked. So, there was a peak and then it came down. There was no not much delay happening here. But here, there is a plateau that happens when the cell reaches a positive potential. So let's talk about what is happening here. Again, there are these three channels. There are the sodium channels, potassium channels, and calcium channels. But what is happening here is, instead of it being a slow influx of sodium, it is a rapid influx of sodium. And the calcium channels are the ones that are acting slowly here. So what is happening is, first of all, in this steep rise at the zero phase, at the zero, the sodium channel. So another thing to note down here is that cardiac muscles also stay at a negative potential of minus 90, which is again very different from how other cells stay. So as we discussed, other cells are mostly present at minus 70. Pacemaker cells are mostly present at minus 60, but these cardiac cells, they are present at minus 90, which is again very different from the other cells. It's unique to the cardiac muscles. So what is happening here is, when the cells are at minus 90 millivolts, the sodium channels, they start opening, and then there is a rapid influx of sodium through these channels, and the cell overshoots zero, and it goes to a positive potential. At this positive potential, as we know, the sodium channels, they close and the potassium channels then open. When the potassium channels open, there is an efflux of potassium happening, and this potassium starts moving out of the cell. And this leads to the membrane potential starting to repolarize, coming again below zero. But what happens at this point is that at some point here, the calcium channels, they open up, and these calcium channels are actually present in the sarcoplasmic reticulum of these cardiac muscles. They are not present in the cell membrane. They're actually present in the sarcoplasmic reticulum. And these channels, they start opening up and the calcium gets released into the cytoplasm. So now two things are happening simultaneously. The calcium is entering the cell, or the entering the cytoplasm, and the potassium is leaving the cytoplasm. So this balances out the membrane potential, and that is why we see a plateau here. That is why it flattens out, and there is no further falling of the membrane potential. But eventually, these calcium channels close when the calcium is all of the calcium is moved out. These calcium channels close, but the potassium is still moving out. as you can see in the phase 3. And since the potassium channels have closed, but the calcium channels have closed, but the potassium channels are still open. That is what returns the membrane potential to minus 90 millivolts again. So what is happening here in this one is that these cells, they stay at this in the phase 2 for a long amount of time, for a extended period of time, as compared to other cells or other action potentials that we saw earlier in neurons, in muscles, or in the pacemaker cells. So this is an important thing to keep in mind, and this is how the action potentials happens in the cardiac muscles. So are there any doubts at this point? Any doubts in this slide, or should we move on? What we saw here is that, first of all, the sodium channels open up. They are again voltage gated. There is a rapid sodium influx. Then the membrane potential rises to around 20 millivolts, which is positive 20 millivolts. This leads to the closing of sodium channels. And then the voltage gated potassium channels open up. Potassium starts moving out of the cell. Cell potential starts dropping. But then the voltage gated calcium channels open up, and the calcium from the sarcoplasmic reticulum gets released into the cytoplasm. And since the calcium balances out the potassium, there is no further drop in the membrane potential. But eventually, the calcium channels close, potassium channels still remain open, and the potassium moves out of the cell, and the membrane potential drops down to minus 90 again. And why is there a plateau? Why is this plateau happening in these cardiac muscles? That is so that the muscles can rest, and so that they actually finish pumping out the blood. Like we saw in the pacemaker cells, this is happening. There is a slow depolarizing potential so that they ensure that the ventricles is empty. Here again, this is happening so that these chambers, they entirely empty out and they pump all of the blood that is present in the chambers. Any doubts at this slide, or should we move on? So, both of these slides were to discuss how the action potentials in the pacemaker cells and in the cardiac muscles are different from each other and how they are happening. Moving on from here, as we looked at the cardiomyocyte action potential, as we discussed, cardiomyocytes, they have a plateau in their action potential. The cells, they remain depolarized for a long period of time. Then we see that this is because sodium and calcium both are present. The cells can stay depolarized only if there is an additional ion in place of sodium. If the sodium ion alone was acting, then the plateau would not have happened. Why is this? Because the gating of the sodium channels is such. It happens at a very narrow window, and the voltage gating only opens when the potential is negative, and as soon as the potential becomes positive, the gating closes and the sodium cannot move inside the cell anymore. And that is when the calcium gets squeezed out of the sarcoplasmic reticulum in a way and leads to the plateau. The calcium needs to be tightly regulated because otherwise it can lead to toxicity in these cells. So these are some pointers to keep in mind. You can refer to these later as well when the slides become available to you guys. Moving on to the different cell types in the cardiovascular system. So can you guys tell me what are the different cell types present in the cardiovascular system? This was discussed during the week. If you have looked at any of the lectures from this week, you would know what are the different cell types that are present in the heart or in the cardiovascular system, not just in the heart. So do you guys know what are the different cell types? RBC, WBC, platelets. Okay, these are the different cell types that are present in the blood. I'm talking about the cell types that form the heart or that form the blood vessels. So basically, what we're discussing here is that in the heart we have pacemaker cells. This is a cell type which are less than 5% of the heart. They form the conduction system. Then we have the cardiomyocytes or the cardiac myocytes. These form the contraction system. Then we have the endothelial cells, which form the walls of the blood vessels or which form the blood vessels. And then we have the cardiac stem cells, which basically are the cells which differentiate into forming either pacemaker or cardiac myocytes. So these are the four different cell types that are present in the heart. Apart from this, there is also the cartilaginous cells that form the walls. So these bicuspid, tricuspid, and the semilunar walls, these are again formed by a different cell type, which is much more cartilaginous than the other cell type because it needs to keep the blood in place, and it needs to you know uh, it needs to fight pressure, and that is why these cartilaginous cells, they form these walls. So these are the five different cell types that are present in the cardiovascular system, and the ones that you mentioned are the ones that are present, the different cell types present in the blood. Okay, so, coming to the last slide, which talks about what are the different sounds of the heart. So, you would already be aware that in our heart we hear this sound lub dup, which is often referred to as dhak dhak in Hindi when you talk about the heart sound. So, but in medical terms, it is recognized that these two different major heart sounds, lub dup, lub dup. Lub, which is also said the S1 sound, it happens when the atrioventricular valves or the tricuspid and the bicuspid, both of them, they close simultaneously. When the atria have emptied their blood into the ventricles and both of these valves, they close shut. That is when you hear lub. As you can see in this image, the atria are empty and the blood is filled into the ventricles. That is when we hear the sound lub. Dub is the sound, or the S2. This is heard when the aortic valves, they close off. When these valves that open into the arteries, these close off together when the ventricles have emptied blood into the arteries. So that is the dub sound. And the time period between S1 and S2, that is between one lub and the dub, is called a systole. That is when the ventricles contract, and this is shorter than the time period between S2 and the next S1. So there is lub dub. So the time period between lub and dub is called the systole, and the between dub and the next lub is called the diastole. This is when the ventricles relax. So this is all about the sounds of the heart. So to give you a view of how this sounds, you can see this is what is happening when it's S1. When it was S1, that was when the AV walls were closing. When it's S2, that is when these two aortic walls close. Okay, so this was all about today, and this brings me to my to the Q&A session for today. So let's answer a few questions so that you have an idea of how these different questions might appear in the quiz or in the exam. So Binakawa is a dash that carries dash. So what is the Binakawa and what does it carry? D part, correct. The vena cava is a vein which carries the blood, which is deoxygenated from the body to the heart. Aorta is the main dash originating from the dash. So you're correct. Aorta is the main artery which originates from the left ventricle and it carries blood to the systemic circulation. What type of cells generate electrical impulses in the heart? You already discussed this. It's a very simple question. Which cells generate electrical impulses in the heart? Correct. So it's the pacemaker cells. We discussed SAAV, bundle of his Purkinje fibers. These are the cells that actually generate electrical impulses in the heart. And out of this, SA cells play the major role because they are the ones that start these electrical impulses. Which of the following chambers receive deoxygenated blood from the body? So we have the four heart chambers. Which of them receive deoxygenated blood from the body? C part, correct answer. It's the right atrium which receives the deoxygenated blood from the inferior and the superior vena cava. Purkinje fibers have dash and are dash than cardiomyocytes and the myocardial cells. So, do Purkinje fibers have more myofibrils or fewer myofibrils, and are they smaller or larger in size than cardiomyocytes or myocardial cells? D part, the Purkinje fibers have fewer myofibrils and they are larger in size. Okay. Moving on, in an EKG, the QT wave signifies what? So in an EKG, what does the QT wave signify? So correct answer, it basically, no, actually the QT wave does not signify the repolarization, it signifies the electrical activity in the ventricles. It's the T wave itself that signifies the repolarization. But the QT segment is not for repolarization, it's for the electrical activity in the ventricles. Which blood vessel carries oxygenated blood from the lungs to the heart? Very simple question. Which of these blood vessels carries oxygenated blood from lungs to the heart? Correct answer, it's the pulmonary vein. What of this is the correct order of electrical conduction in the heart? Which one of these is the correct order? B part, you're correct. So basically, the electrical conduction, as I said, originates in the SA node, moves on to AV node, bundle of His, and then the Purkinje fibers. Which heart valve separates the left atrium and the left ventricle? Correct. It's the C part. It's the mitral valve, also called the bicuspid valve. Which layer of the heart is responsible for contraction? Which of these layers is responsible for contraction? So myocardium is the correct answer. Yes, the myocardium is basically responsible for the contraction of the heart. Which of the following is not a part of the heart's conduction system? A part, correct. AS junction is nothing to do with the heart's conduction system. It's the SA node, AV node, bundle of His, and Purkinje fibers. Which of the following in the heart's conduction system is the primary pacemaker and generates the initial electrical signal? I've given away this answer multiple times. Yes, the correct answer is the SA node. It's the primary pacemaker generates the initial signal. Which part of the ECG corresponds to ventricular depolarization? Correct. It's the C part, the QRS complex. The lub sound of the heart is caused by what? Which of these causes the lub sound of the heart? Correct. So the correct answer is C part, closing of the atrioventricular valves. Right. Which is the primary function of the left ventricle? What does the left ventricle do primarily? Correct. So basically the left ventricle is the one that pumps blood to the body. And it's the right ventricle that pumps blood to the lungs. Which of the following is not a type of the heart cells? Correct. So the correct answer to this is neurons. We heard that there are pacemaker cells, there are these contractile cells which lead to contraction, endothelial cells in the blood vessels, but neurons. Uh, Sorry, the answer here is incorrect. But neurons is the correct answer. They are not a type of the heart cell. What does the P wave on an ECG represent? Correct. It's the atrial depolarization. The dub sound of the heart is caused by A part, closing of the semilunar walls. Correct. Which of the following is a common cause of an abnormal heart sound? So you would have heard of these abnormal heart sounds, which are also called heart murmurs. So what are these mostly caused by? What leads to a heart murmur? You can think of this by thinking of what is it that actually causes a heart sound, and then what would it be that leads to a heart murmur? Correct, B part. Since heart sounds are actually they're actually because of the closing of these walls. If these walls dysfunction, they lead to a heart murmur. The T wave on an ECG represents what? So what is the T wave for in an ECG? Its correct answer is ventricular repolarization. Thank you for joining today's session. If you have guys have any doubts about this week's lectures, you can ask me. If everything is clear, then thank you for joining. This was all for today. You can exit the class.